Okay, welcome everybody to the September session of Amplifying Voices. It's good to be back after I think quite a long summer without any speakers. We're very excited to have two great talks today by some ECRs. So yeah, this is Amplifying Voices. We are the ASLO Early Career Committee. My name's Kale, and I'll be the moderator. So how this morning is going to work is we've got two great talks. We're going to have the first one, which you can see on the screen, presented by Yash. He'll be going first. Unfortunately, we're having a little bit of technical difficulty. Yash is in the room, but we're having some trouble with the audio visual. So we're going to play the talk. And then if you have questions, you can put those questions into the chat to make sure that he can see them. And then after his talk, he will answer some of those questions in the chat. And then following that, we will have our second speaker, Maja, but we'll get to her talk after Yash. So Yash is an early career researcher pursuing his doctoral studies in the India Institutes of Science Education and Research in Kolkata. He's investigating the positive impacts of nature-based solution approaches towards sustainable coastal aquaculture, which is something we're going to be hearing about today. He's interested in exploring the sustainability of the blue economy at that nexus between science and society, and he has worked in both freshwater and marine ecosystems. And today we're going to listen to his talk about these mangrove leaf litter dynamics in aquaculture ponds. So I'm going to start or get Karen to start the video, and great.
Hello everyone, I am Yash from Indian Institute of Science Education and Research, Kolkata, India. Today I am going to talk about my ongoing research that mangrove leaf litter dynamics in the integrated mangrove aquaculture ponds of Sundarbans, synergy between the phenolic compounds and nutrient pools for sustainable blue economy. The blue economy is a prime focus in present scenario, both regionally as well as globally, to achieve the United Nations Sustainable Development Goal 2030. The main agenda behind this goal is to break down the economic barriers between least developing nations and developed nations, and bring down together on a platforms where we can think about the boosting the ecological sustainability and think about the sustainable approaches for revenue generation with better livelihood opportunities. Globally, blue economy is estimated at approximately a 2.5 trillion value per year USD dollars, and it provides approximately 30 billion job opportunities globally, and it contributes about 3% of the global GDP presently. In part of Asia, particularly South and Southeast Asia, has the major focus as blue economy accounts almost 20% of the GDP. And among Asian countries, India holds second rank in aquaculture production, and 4% of the India's GDP is dependent on blue economy. When we look at the share of aquaculture, in year 1986, it was 23%. Now by 2030 we have to increase it by 62%, whereas in export also we have to increase it by 61% by 2030. And so the state like Andhra Pradesh, West Bengal and Gujarat are major players in export. When we look at the coastline, length of the coastline of India, it is almost 8,000 kilometers, where West Bengal holds 8,000 10 kilometers with exclusive economic zone, as well as the Bay of Bengal represents large marine ecosystem with annual value of fishes production is 3.4 billion USD dollars. When we look at the export performance of marine products from India, in year 2010-11, it was around 3,000 million USD dollar, whereas in year 2021-22, it has just doubled and it is now almost 8,000 million USD dollars. And when we look at the item-wise exports, our frozen shrimp contributes almost 70% of these values, and we are the leading exporter of frozen shrimps, and we export our materials with countries like USA, China, European Union, Southeast Asia, as well as countries like Japan and Middle East. So we have to think about the sustainable approaches through which the productivity of aquaculture ponds can be increased. Is here mangrove plays a significant role. All the integrated mangrove aquaculture practices is a green approach of aquaculture, where the mangrove plants are planted on the dikes of the aquaculture farm, as well as in the middle with the formation of Iceland. The litter derived from mangrove plays a significant role in balancing the nutrient stoichiometry, as well as these litters has also antimicrobial properties which enhance the yield of the shrimps and other bioresources yields. What really happens with the mangrove litters? The integrated mangrove aquaculture ponds, where the mangrove plants are present, provides a canopy cover, which result in a favorable environment for the production of tannic and gallic acid from the litter degradations, and further leaching of these phenolic compounds chews up the dissolved organic carbon as well as dissolved nitrogen pools, whereas in the open estuarine ecosystems, the tannic and gallic acid production is not happens due to the broad light. So the tannic and gallic acid gets exchanged through the hydrodynamic exchanges or from runoff, and these litter phenolic compounds, tannic and gallic acid, contribute a significant role in carbon and nitrogen pool, as well as it contributes almost 30 to 60% of the total primary production in estuarine ecosystem. When we look at the Sundarbans is a UNESCO World Heritage Site as well as a Ramsar site, and in the area of Sundarbans, prominent aquaculture practices are being done through the cutting down the fringes of mangroves, through cutting down the mangrove plants and developing it in aquaculture ponds. We are well aware of the fact that the pollutants which are generated from the aquaculture ponds as the commercial feed, and a lot of antibiotics are being used for the shrimp cultivation, and these result in the pollutants which are ultimately released in the nearby ecosystem and affects the biodiversity of this coastline region in the Sundarbans, in the area of Sundarbans. So why Sundarban is important? When we look at these pictures, we can clearly see this provides a livelihood opportunities for millions of people. As you can clearly see that boats are ready for fishing activities, whereas the family members are busy in fishing, whose livelihood is directly dependent from the yield which they captures from the fish which they captures from this region, as well as the people that are extensively doing aquaculture practices. And here in the picture they are showing the yield of shrimps from their pond. Or why this strategy is important. When we look at the spatio-temporal expansion of aquaculture areas in Sundarban, in last 2 decades, you can clearly see how the ponds number has increased in the region of Sundarbans through the demarcation of yellow points, and it has just doubled in the area length. And the two districts of West Bengal, that is North 24 Parganas and South 24 Parganas, are being used for extensively used for the same farming, and this region also has the mangroves covered. So we have to think about how we can increase the productivity to meet our nation goal as well as United Nations Sustainable Development Goals. We possess the best ecological dimensions with large export market with multiple countries, as well as we have the government schemes like MISTI, which can help us, which has the motto of Mangrove Initiative for shoreline habitats, as well as to generate the tangible incomes from this. So based on this, my broad objective was to quantification of mangrove leaf litter derived tannic and gallic acid in the IMF ponds, and how this mangrove litter derived tannic and gallic compounds is affecting the nutrient stoichiometry, as well as how investigate the role of tannic and gallic on the bioresource yield of these IMF ponds. Based on this, I selected total number of 12 aquaculture ponds in the region of Sundarbans, where row sampling are being done and on the monthly basis. Here we collect the samples from each of the aquaculture ponds and do the measurement of essential parameters like pH, dissolved oxygen, and other parameters which are important for the shrimp cultivation and other bioresource yield cultivations, and further we bring down these samples in the lab. Here we look at what are the biological communities present in this aquaculture ponds, as well as we do the other different parameters measurement like dissolved nutrients and the metal concentrations present in these ponds. So when we look at the spatio-temporal dynamics of this phenolic compounds, that is this tannic and gallic acid, we found that the concentration of tannic and gallic acid was higher in the ponds which are integrated aquaculture ponds, and you can clearly here see in the green plots. The green plots are representing the ponds which are integrated mangrove aquaculture ponds, whereas the red plots are showing the ponds which are non-integrated mangrove aquaculture ponds. Further, we look how this tannic and gallic acid is affecting the important parameters like dissolved oxygen and pH. Here we found that there was no negative effect of the tannic gallic acid on the concentration of dissolved oxygen, as it was found that it was in the range of 5.4 to 6.3 milligram per liter, and no hypoxic condition was, as it reveals that there was no hypoxic condition in any of these ponds, whereas pH also ranged between 7.3 to 8.8, which is favorable for the shrimp farming. When we look at the dissolved nutrient concentrations, here also we found that the ponds which are integrated mangrove aquaculture ponds, it has the higher concentration of dissolved inorganic nitrogen, dissolved reactive silicate, and dissolved organic carbon concentration. And in these ponds also the buffering capacity was balanced, as the alkalinity value shows this. Interestingly, when we look at the dissolved orthophosphate concentrations in the ponds which has the integration of mangrove, that is integrated mangrove aquaculture ponds, the dissolved orthophosphate concentration was also higher in these ponds, and it was balancing the rate field ratio, which is one of the important parameters for the primary producer communities. Further, we look at we look at the concentration of the pigments, that is chlorophyll A, which is a proxy for the total abundance of the primary producer community. We found that the bond in the integrated mangrove aquaculture bonds, the chlorophyll A concentration was also higher, and carotenoids value concentration were also higher in these bonds. That means that the phenolic compounds, tannic and gallic acid was boosting the growth of primary producer, and through the nutrient balancing, as well as it is also positively resulting in the high yield of the bioresources present in these ponds like shrimp and other fishes. Further, we try to look at the microbial communities which are helping in the degradation of these mangrove litters present in these ponds. So that we saw we look at the prokaryotes files at class level. Here we found that presence of bacteria was high, as well as the actinobacteria's abundance was also high, and these all were playing a significant role in carbon nitrogen cycling, as well as in the litter degradation. Whereas when we look at the eukaryotes class level, here also we found there were the abundance of phytoplankton communities and blue-green algae like Coscinodiscophyceae, Basilerophyceae were high. And the functional property, these microbial communities were playing a major role, as it is revealed that these were helping in the amino acids metabolism as well as carbohydrate metabolism in these bonds. So what I saw in the field, I tried to look at it through a microcosm experiment, that whether the litter-derived phenolic compounds, tannic and gallic acid, are playing any role in balancing the nutrient chemistry. Based on this, my research question, where does the mangrove litter defines the chemistry and nutrient stoichiometry of nutrient stoichiometry, and does different sources of tannins are responding differently. Based on this, my objective is also to quantify the tannic and gallic acid in an enclosed system over temporal basis, and monitor the consequences of this phenolic compound on breakdown on the essential nutrient chemistry, as well as look at how these are affecting the rate field ratios, that is NH2P or SI/N ratio, and predict the shifting of the primary producer in an enclosed system. For this, I designed a microcosm experiment. 6 containers were chosen. In that, the water samples were brought from 2 IMA ponds, and it was kept over a time for 21 days. And we tried to look at the breakdown of the tannic and gallic acid, and in 2 containers we put the synthetic tannic acid also to check if the sources of tannic acid is also playing a major role or not. So what we saw here, that with the days, there was the breakdown of tannic acid, and which resulted in the increase of gallic acid concentration in each of the containers on temporal basis. Whereas we also found the episodic changes in the dissolved inorganic nitrogen profiles with the days, as the gallic acid, further breakdown of gallic acid were resulting in the increase of the dissolved inorganic nitrogen concentration. Interestingly, when we look at the dissolved orthophosphate profile, here also the dissolved orthophosphate concentration was increasing in all the containers, and the episodic changes in the concentration was observed. And the most interesting thing, we found that with the time, there was a decrease in the concentration of dissolved reactive silicates, which which shows that there was the limitation of silicate with the time, and it was affecting the N is to Si ratio, and there was with the limitation of the dissolved silicate, there was shift in the primary producer communities from diatoms. It was shifting towards the green algae as the ratio N is to Si ratio were less than 1 in all the containers. But we did the statistical analysis to look at how the gallic acid is affecting different environmental parameters. Here we found there was a statistically significant positive correlation between gallic acid and dissolved orthophosphate, and also with the dissolved nitrate. So the finding which I have drawn till now, that this tannic and gallic acid was not creating any hypoxic condition in any of the ponds, as DO range between 5.4 to 6.3 milligram per liter. Concentration of tannic and gallic acid was also higher in the integrated mangrove aquaculture ponds, and dissolved nutrient concentration was also higher in the integrated mangrove aquaculture ponds. In parallel, DOC pool was also higher in the integrated mangrove aquaculture ponds. That means that this tannic and gallic acid is balancing the nutrient stoichiometry in these ponds, whereas the microcosm experiment also shows that the breakdown of tannic acid result in the increase of gallic acid concentration, where further breakdown of this gallic acid is resulted in the increase of dissolved inorganic nitrogen concentration with the experimental time frame, whereas the dissolved reactive silicate decreased over the duration, and dissolved phosphate, which shows the episodic positive and negative variation with the duration of the time. That means overall conclusion is that this the mangrove integrated mangrove aquaculture practices is the sustainable way through which we can balance the nutrient stoichiometry for sustainable growth of shrimp and other bioresources, and this can lead a sustainable practices of aquaculture in the region and result in a less generation of pollutants, and which are majorly generated from the aquaculture practices. And these mangroves later can also play the role of antimicrobial properties, and which can decrease the diseases which are spread amongst the shrimps in these ponds. I would like to acknowledge the audiences which are listening me patiently, as well as I would like to acknowledge integrated taxonomy and microbial ecology research group members, my research group members, for supporting me with research ideas and in my research work, as well as I would like to thank ASLO Kolkata for providing me all types of facilities, and I would also like to thank ASLO for providing me the platform to present my research. Thank you. If you have any queries, or you can email me and text me. Thank you, everyone.
Great, thank you, Yash. As I said before, if we have any questions for Yash, he is here, but may not be able to speak. So if we can put them into the chat, that would be ideal. You have a couple minutes to do that if anyone has any questions. Okay, we have one question in the chat from Hans Peter. Question is, How do you derive activity from bacteria, community composition, or taxa? Yash, if you can see that or heard that, can you reply in the chat? Don't know if Yash is typing or has seen this. If not, if he has, maybe we'll get the response. And if not, maybe Hans Peter, you can contact Yash directly at. Oh, here we go. We have a reply from Anwesha. The research group uses metagenomics to predict the pathways and then validate using RNA. That she will get details back to us. Great. Okay. Thank you again to Yash. We're going to move on to our second speaker, Maja Ilić. So if you want to share your screen. Maja's a postdoc at the Swiss Federal Research Institute in Switzerland. And before that, she had done her PhD at the University of Cologne in Germany and spent some time as well as an analyst at Queen's University Belfast in the UK. Today, she's going to talk to us about her work on these dietary fatty acids and how they relate to Daphnia populations. So, Maja, please start when you're ready.
Yeah, sorry, I was just struggling a little bit with unmuting myself, but thank you so much for the introduction, Kale, and also thank you everyone for being here. Thank you for the opportunity to present here the work that I've done mainly in my PhD thesis, which I finished in 2019. So sorry for those who have already heard about my talk here, but it's still nice to speak about it again because it's still very relevant. So I'll be talking about dietary fatty acids and how they affect the trophic transfer efficiency and Daphnia population dynamics in freshwater ecosystems. So I have been there working with Daphnia. Daphnia, as you may know, the water fleas have a key role in freshwater food webs. They feed on phytoplankton, which is on the bottom of food webs, but they also serve as a food for the planktivorous fish, for example, and some of the invertebrates such as herbivorous larvae. That means that also they are responsible to basically transfer the energy and nutrients from these lower trophic levels to the higher trophic levels in the freshwater food webs. Daphnia themselves are unselective filter feeders, which means they do not select the food particles that they eat. They pretty much filter everything that is there and that fits through their Carpox gap. They are capable of cyclical parthenogenetic reproduction, which means that females don't necessarily need males and they can just clone themselves in optimal conditions. And their genome has been sequenced, which basically makes this Daphnia a really great model organism to study ecoevolutionary dynamics. Some of the factors that determine the dietary quality of the phytoplankton for the zooplankton, including Daphnia, are on the one hand, the elemental stoichiometry, specifically looking into the ratio of the nutrients carbon, nitrogen, and phosphorus. And on the other hand, the biochemical composition, for example, the presence and the content of dietary fatty acids. These fatty acids are crucial for a variety of physiological functions, specifically in animals as well. However, the omega-3 and omega-6 polyunsaturated fatty acids are mostly synthesized by primary producers, in this case by the phytoplankton, and they cannot be de novo synthesized by the zooplankton, or even if they are synthesized, it usually happens at a very, very low rate. And this makes these polyunsaturated fatty acids essential nutritional compounds for the zooplankton, such as Daphnia. So some of the most common and quite studied polyunsaturated fatty acids are alpha-linoleic acid, shortly ALA, which is a short chain omega-3 fatty acid that has been shown in the field studies, but also in the lab, increases the growth rate of Daphnia, the more of ALA is present. Another one, which is probably the most recognized polyunsaturated fatty acid in this case, is the icosapentaenoic acid, shortly EPA, which is a long chain omega-3 fatty acid, also shown in the field and in the lab to increase the growth and also the reproduction of Daphnia. And the third fatty acid I will focus today is arachidonic acid, shortly ARA, which is a long chain omega-6 fatty acid, so different omega family in this case, which has been shown in the past not to increase the growth of Daphnia. But however, in some of my studies, I actually showed that this is not the case, and it depends on the content that is supplemented to the Daphnia and also on the Daphnia species. So in this case, I conducted growth experiments with increasing concentration of EPA and ARA, so the two long chain PUFAs. And here I used the different Daphnia species. On the left, we see Daphnia pulex, which is of a middle size, and on the right, Daphnia magna, which is of a large size. And we see that in both cases, with the increase in concentration, we also see this increase in the somatic growth rate that actually kind of follows this saturation curve. And what we could calculate from the saturation curves that we did for the somatic growth rate, but also for the reproduction, basically the clutch size, we can then calculate the maximum possible growth rate and reproduction if we assume that we have unlimited availability of these fatty acids. And if we compare these numbers, so on the left, we have the asymptotic growth rate, and on the right, the asymptotic clutch size, basically the number of eggs. We see that between the two fatty acids, which are here shown in different colors, green for EPA and blue for ARA, we do not find a significant difference. But between the species, we do. So we see that, for example, Daphnia magna, which is shown more on the right side, which is the largest species, also has the potential to reach much higher somatic growth rates, significantly higher compared to Daphnia pulex, and also to reach significantly higher and larger clutch sizes, number of eggs. So with this, I could show that actually, other than assumed before, ARA and EPA are equally relevant for the fitness of Daphnia. In my other work, I isolated 100 Daphnia longispina individuals. These are of even a smaller size than the two Daphnia species used before. And these individuals were isolated from a natural plankton population of the Lake Klostersee, which is situated in Upper Bavaria in the south of Germany. I then used microsatellite analysis to identify the different genotypes and could then culture 12 different Daphnia longispina genotypes. And afterwards, I conducted standardized growth experiments where, again, I used a green algae that was supplemented with PUFAs, in this case, ALA, EPA, and ARA, so the three PUFAs I introduced earlier, and then looked into the growth rate and susceptibility of the different Daphnia genotypes to the presence of these PUFAs. As mentioned, I calculated the susceptibility, which is calculated as the difference of the growth reached in the PUFA-free diet, basically the control treatment, minus the growth rate in the PUFA-enriched diet, divided then again by the growth rate in the control treatment times 100. And here you would expect negative values for this because we are expecting a higher growth rate if PUFAs are supplemented to the diet. And what we can see here in all three panels representing the different PUFAs supplemented, the different colors then represent the different genotypes. We see that we find differences between these genotypes in how susceptible they actually are to the presence of these fatty acids. So with this second study, I could show that there is indeed an intrapopulation variability in the susceptibility of these Daphnia genotypes to the PUFA limitations. So if we now imagine a more natural system, we just imagine a lake. And this lake, for example, has a certain diversity of phytoplankton, of the primary producers. And this community, again, offers a certain diversity of the fatty acids that are present. And then at the same time, we imagine that we have a certain Daphnia population that consists of different genotypes that all coexist with each other. What would happen if we experience biodiversity loss at the level of primary consumers? Primary producers. And important here is to mention that the PUFA content is actually taxon specific, which means that certain PUFAs are only produced by certain phytoplankton groups. So what we expect here to happen is that if we lose some of the diversity in the phytoplankton community, we might also lose some of the fatty acids. And then again, if this does happen, the question is how this would then affect the population dynamics and the genotype frequencies in the Daphnia population. So from this, I hypothesized that the loss of diversity at the level of primary producers, in this case, the phytoplankton, might lead to a loss of traits. And with trait, I here mean the content of these PUFAs and their availability in the system, which would then lead to an altered PUFA availability. And then together with the intrapopulation variability and susceptibility that I've shown in the study before, I hypothesized that there is going to be a joint effect on the intraspecific competition of Daphnia. So to address these hypotheses, I conducted a common garden experiment, basically a Microcosm experiment. And here I used basically phytoplankton, a natural phytoplankton community originating from the same lake in Germany, the Lake Klostersee, from which we also isolated the Daphnia longispina genotypes. And I used different volumes of inoculate to basically make new communities and expected to reach some kind of a diversity gradient that should, so with diversity increasing with the size of this inoculum. And then I used these pre-grown diversity manipulated communities to then inoculate the bigger mesocosms. And in total, each of these pre-grown communities was used 3 times. So we had 15 mesocosms. Each mesocosm was filled with 60 liters of water, and they were spread across 2 different pools to be able to control more for the temperature, because the experiment took place over a course of 98 days in the summer in 2016, I think was it. This was then the start of the official growth phase, which then lasted for 4 weeks and was entirely Daphnia free. After the 4 weeks, we then added Daphnia to the system. And here we picked 3 different Daphnia genotypes from the study before that showed these pronounced differences in their susceptibility to the PUFA limitations. And of each of these genotypes, they were all synchronized. They were all from the same clutch. We then added 20 individuals, in total 60 individuals of Daphnia to each of these mesocosms, resulting in a concentration of 1 individual per liter. And this was then the official start of the grazing phase, which then lasted for 10 weeks. Throughout the entire experiment, we took weekly samples of the Daphnia and of the water, basically the phytoplankton. We measured quite a few parameters, of which I will only show a few today. For example, chlorophyll A, which we use as a proxy for the biomass of the phytoplankton community. Then we also look into phytoplankton community composition and into the fatty acids. The way I did this was that I first extracted and identified the pigments from the phytoplankton community using the high pressure liquid chromatography. And then from this pigment composition, I was able to then estimate the phytoplankton community composition by using a matrix factorization program called ChemTux that takes into account the total amount of all these pigments and the pigment ratios of the groups that I expect to find in the community. And at the same time, I extracted and identified the fatty acids using the gas chromatography. From the fatty acid composition and the phytoplankton community composition, I then calculated the Shannon diversity indices. And here I calculated the ChemTux diversity, basically the diversity of the phytoplankton community, and then also the diversity of the fatty acids, including all fatty acids found, and also including only the polyunsaturated fatty acids. As for the Daphnia, I was here counting how many of the Daphnia we find every week, and also again use the microsatellite analysis on a subsample of Daphnia each time to then basically calculate the relative abundances of the 3 different Daphnia longispina genotypes. So as mentioned earlier, the experiment lasted in total 98 days, so 14 weeks. Now here on the left, we see the chlorophyll A, the proxy for the biomass of the phytoplankton community and the dynamics over time. And on the right, we see the abundances of the Daphnia longispina individuals given in individuals per liter. Gray is always the growth phase, which was Daphnia free, and black is always the grazing phase where we added the Daphnia. And what we find here are some typical producer-consumer dynamics. So we see that in the growth phase, which was Daphnia free, there was an increase in biomass across all these mesocosms, here shown in different colors, representing the different inocula we use at the beginning. And then as soon as we add Daphnia, obviously the phytoplankton community, the biomass goes down due to the grazing pressure. And this goes down quite a lot. And then at some point, you also see the Daphnia abundances go down as well, obviously, probably because of the lack of the food. And in that moment, when Daphnia abundances decrease, we again see a recovery of the phytoplankton biomass somewhere around day 77, which then increases until the end of the experiment, while Daphnia abundances decreased. In the following slides, I will always focus on day 28 when it comes to phytoplankton, because these were then the official starting conditions of the entire experiment. And for Daphnia, I will focus on day 56, basically 4 weeks after we added Daphnia, which corresponds to approximately 2 generations, including the 3rd clutch of Daphnia at optimal conditions. On the left, we now see the phytoplankton community composition on day 28. Each bar is representing one of the mesocosms, and these are now ordered along an increasing diversity. So the white dots are showing the phytoplankton diversity, the Shannon index. And what we can clearly say is in every mesocosm, we actually found quite a lot of chlorophytes, the green algae, but we also had a few mesocosms that had some dinoflagellates and some chrysophytes. But it's really obvious that the green algae were dominating the community in these mesocosms. On the right, we see the fatty acid composition here subdivided into 4 groups, purple being the saturated fatty acids, blue being the monounsaturated fatty acids, and then in green and in yellow, the omega-3 and omega-6 polyunsaturated fatty acids. Again, bars are representing the mesocosms, here ordered in the same order as the mesocosms on the left. We do not clearly see here that there is actually a relationship, but if we do plot these diversity indices together, we do find a linear positive relationship between the phytoplankton diversity here on the x-axis and the fatty acid diversity on the y-axis. And if we do the same, but use the diversity of PUFAs only, we find again a positive relationship, which is even stronger. So it has an R-square here of 0.4. And with this, I could actually show that the loss of diversity in the phytoplankton community does indeed lead to a loss of traits, in this case, to a decreased diversity of the fatty acids and also the polyunsaturated fatty acids. When it comes to the Daphnia longispina genotypes, here I'm showing the dynamics of the 3 different genotypes over time. We see on the left the genotype KL14 didn't really do well. It decreased pretty much immediately after it was added, and it didn't really recover throughout the time. And then if we look at the 2 other genotypes, KL83 and 93, we see that there is a little bit of happening here. So we see some increases and some decreases, but by the end of the experiment, it seems like they are basically somewhere in the middle. And we can look at specific days here. For example, the day 56, as mentioned, 44 weeks after the start of the experiment. Here we see that the KL93 was mostly dominating the communities here. If we then look at day 77, we actually do not even find any of the KL14, which was probably just a sampling error. But what we actually find here is this 2-to-1 ratio of the genotypes KL93 and KL83 across these mesocosms. And then at the end of the experiment, as mentioned earlier, we see that actually KL83 recovers, which was visible in the plots before, So here there is actually the question if you would run this experiment for a long time and if the system would obviously work out, if we would actually find this coexistence of these two genotypes where their relative abundances basically just like go up and down a little bit, but in theory they just coexist over time. So lastly, what I'm showing here is a correlation matrix where I'm now comparing the relative abundances of the three genotypes with the relative abundance of some of the most important PUFAs. Here I also included the DHA, which is another omega-3 polyunsaturated fatty acid, and also the diversity of these PUFAs. And if we now only focus on what is really most important, which would be kind of the relationship between the Daphnia and the PUFAs, what we find here is that the genotype KL14, the one that really performed very badly, its abundance didn't really show any relationship to what PUFAs are available. But if you look at the two other genotypes, the ones that seem to coexist, we see that KL83, for example, shows a positive correlation with the diversity of PUFAs and also positive correlation with the abundance of ARA, which is the omega-6 polyunsaturated fatty acid. And then also what we find is a negative relationship of KL83 with the abundance of ALA, while on the other hand, we find a positive relationship of the other genotype, the KL93, with the abundance of ALA. So to sum this up, I could show that the loss of diversity within the phytoplankton community indeed leads to a loss of traits, resulting in an altered PUFA availability. And together with the intrapopulation variability and susceptibility to these PUFA limitations, we actually could show that there are some effects on the specific competition of Daphnia. And with that, I conclude that the PUFA content is actually a phytoplankton functional trait that determines the trophic transfer efficiency, but also the Daphnia population dynamics in freshwater ecosystems. And with that, I would like to thank my supervisor, my PhD supervisor, Patrick Fink from the Fink Lab, our collaborators, Maria Stockenreiter, Herbert Stibo, and Eric von Ilić, who have been a really great support throughout my entire thesis. Obviously, however, my true heroes are the Daphnia and all the students that were very helpful throughout this entire time. All of these experiments were quite huge and would have not been possible without them. And I thank you all for your attention.
Thanks very much, Maja. Great talk. We definitely have some time for some questions if anyone has any for Maja. Yes, Hans-Peter, go ahead.
Okay. I have a quick question. So concerning your conclusions, the third point that the PUFAs or the fatty acid pattern indeed determines the presence of certain Daphnia genotypes. Can that be really derived? I mean, this step is not fully clear to me because in nature you have a lot of other factors maybe also impacting the availability of fatty acids and other food. I mean, you see something in the experiment, I agree, but can it be translated to natural conditions?
I thank you for the question, Hans-Peter, by the way, great to see you. I would still say so. I would still say that even if it's maybe not entirely visible in the experiment that I did, I could imagine that even if you go a bit larger, let's say if you do this in a lake or anything, that you would definitely find changes in your Daphnia community that might go back to what is available in terms of PUFAs. We did also look into other parameters such as the stoichiometry, but for example, there we didn't really find much, to be very honest. It was kind of pretty much all over the place. But with these PUFAs, I mean, we've seen it also in the experiments. First of all, we do find really these huge differences between the Daphnia. And we also expect that some of this might be due to how they relocate their resources, if they put more into growth, more into reproduction, if some of them are maybe capable of some of the biosynthesis, for example, and if so, if this rate, for example, also differs between the genotypes. I will still say that the community that is present, is it either due to whatever is present in terms of phytoplankton species, maybe their palatability or anything, or if it's the fatty acids. It is obviously hard to separate this, at least in our experiment, but I would definitely say fatty acids play a role. If it's as strong as I'm trying to sell it, that's definitely a fair question, but I do think that they are responsible for at least some of the dynamics that we see. Absolutely.
Great. Karen also has a question, it looks like.
Yes, Maja, thank you for that great talk. That's super interesting to me, who is not that into these things. But maybe adding up to Hans-Peter's question, or going even to a higher or lower level, I don't know. Like when it comes to nature conservation efforts, what would that mean? What do your results imply? Like, how can we protect?
Mm-hmm. So the thing is that in this case, like really strongly focus kind of on like what the diversity loss would mean for the population dynamics without addressing actually how much biomass is produced, right? I would argue that if you imagine that you have a strong diversity loss, which might actually affect strongly, let's say, all these groups such as dinoflagellates, because it feels like green algae, they kind of always come through, right? And green algae, however, are of kind of a intermediate food quality. They produce ALA in quite some big amounts, but they mostly do not have any of these longer chain polyunsaturated fatty acids. So what I would assume here is not only that we would obviously find these changes in the Daphnia populations, but it also, the Daphnia would probably just grow less. We would just produce less of this mass and less of the really good quality food. And then looking into the higher levels, looking into the aquatic invertebrate larvae, looking into fish, again, all of this will probably be affected in some way in terms of food quality. I mean, we've heard of omega-3 being important for us as humans as well, and we can get them from fish. Well, fish does get it from the phytoplankton over the Daphnia. So basically, what we would need to do is just really try to conserve the phytoplankton as such, because that's where kind of this food quality part really comes from. And then really look into preserving all of these species and trying to get more than just the green algae that you will probably find everywhere and all the time.
All right, thank you.
Great. Do we have any final questions?
Question in the chat, Kale.
Yeah, there is one from Mina.
Yes, from
Yeah,
Mina, yes.
I'm just reading it. Hi, Mina, great to have you here. Think about PUFA preference independence in. Yeah, so unfortunately, me and bacteria are very bad. But I'm pretty sure that there is some research done on that, or it's at least ongoing. And also, and this is something that I just figured out recently, that when it comes to probably metabolism or even to the fitness of Daphnia, that actually we should look into combined effects of presence of PUFAs and how the microbiome in the gut looks like. I'm pretty sure that there might be something that is worth looking into, but I'm unfortunately not really the person to ask. But absolutely something that should be kind of, part of some future research where we kind of combine this. Yeah.
Great, thanks. Don't see any other questions, any other hands. So maybe let's thank Maja again, and Yash, who's already left, but thanks Yash and absentia, and thanks to all of you for coming to the session of Amplifying Voices. Our next one we'll be announcing soon, should be the end of October, and we'll keep you all posted on the ASLO social media and emails. So thanks again. You'll have a good rest of your day.
Bye-bye.
Yeah.